STIM2, who, IM2, STIM2, who. Stromal interaction molecule 2 STIM2 is a protein that in humans is encoded by the STIM2 gene. This gene is a member of the stromal interaction molecule STIM family, which comprises only two members together with its homologue STIM, and likely arose from a common ancestral gene. They encode type 1 transmembrane proteins that are located in the sarco/endoplasmic reticulum SR/ER into the cell. Alternative translation initiation from an AUG and a non-AUG UUG start site results in the production of two different STIM2 isoforms. Both members of the STIM family were identified in 2005 as free calcium Catwall plus sensors which participate in a mechanism of Catwall plus entry into the cell referred to as store-operated Catwall plus entry SOCE. Many cellular processes and signaling pathways are started by previous release of Catwall plus stored in subcellular organelles, which needs of a continuous refilling. SOCE is considered the mechanism of store refilling and an essential mechanism of Catwall plus signaling in non-electrically excitable cells. While STIM triggers SOCE, research on STIM2 function suggests a major role as feedback regulator. STIM2 detects small decreases in Catwall plus content stored in the S/ER, switches to the activated state and interacts with so-called store-operated Catwall plus SOCE channels located in the plasma membrane, such as Orai or TRPC channels, allowing SOCE. Although the functional role of STIM2 has been elusive for many years, studies performed in 2009–2010 on murine models suggested that STIM2 participates in processes of the development and functioning of many cell types, including smooth muscle myoblasts, cells of the immune system, and neurons, and is involved in gene. In 2001, STIM2 was identified as a new human homologue of the STIM gene representing the second member of a two-gene family in vertebrates. The STIM2 gene contains 12 exons and 11 introns located on the human chromosome 415.1 and on the large arm of the mouse chromosome 5, close to the centromere. The members of STIM family most probably have evolved from a single gene in lower multicellular eukaryotes into two related genes in vertebrates, since human STIM and STIM2, as well as Drosophila melanogaster STIM have a conserved genomic organization. The D-STIM protein of 570 Os exhibits equal similarity to both STIM, 33% identical, 50% of amino acid sequence conserved, and STIM2 31% identical, 46% of amino acid sequence conserved. Unicellular eukaryotes such as Monocyga brevicalis, a unicellular chonoflagellate, has been reported to have a STIM-like gene. However, no STIM-like genes have been identified in prokaryotes. No additional STIM-like proteins have been identified until now in vertebrates. Protein structure. STIM2 protein is a type I transmembrane protein located in the S/ER. Human STIM2 consists of 833 amino acid residues Os 105-115 kDa 148 additional Os compared to human STIM. Their N-terminal regions share 66% similarity over 577 Os, 85% of the amino acid sequence of STIM. Only the extreme of the C-terminal region shows a significant sequence divergence. The domain architecture of both isoforms is highly conserved in vertebrates. Fig. 1. Mouse STIM2 shares a 92% identity with human STIM2 in the aminoacid sequence according to the pairwise alignment generated by BLAST. Their domain structure is also highly conserved. Fig. 1. Human STIM2 is post-translationally modified in vivo, such as maturation by cleavage of N-terminal S/ER signaling peptide 14 Os, glycosylation, and variable degrees of phosphorylation, but the phosphorylated sites are still unknown. Fig. 1. Domain architecture. The N-terminal region of STIM2 is located in the S/ER lumen and contains a canonical EF-hen catwall plus binding motif, a hidden EF-hen catwall plus binding motif discovered recently, and a sterile motif SAM domain, a well-known protein-protein interaction motif Fig. 1. The N-terminal portion is separated from the C-terminal region by a single-pass transmembrane motif that is highly conserved in all STIM proteins. The C-terminal region contains a high degree of helical structures. A large proportion close to the transmembrane domain comprises a region similar to an esrin/radixin/mosin ERM domain that contains two coiled coil domains. The coiled coil domains mediate interactions between STIM proteins, allowing them to bind each other and form homo and heterodimers Fig. 1. Finally, further towards the C terminus, STIM2 contains a proline/histidine-rich motif and a lysine-rich tail of 17 Os Fig. 1. EF-hand SAM region. Since the EF-hand and SAM-EF-SAM domains are vital to STIM function and SOCE regulation, they are now discussed in detail. The EF-hand domain is a catwall plus sensor used by STIM protein to detect changes in catwall plus concentration inside the S/ER. STIM isoforms become activated when catwall plus bound to the EF-hand motif is released as a result of a decrease in catwall plus levels inside the S/ER store after IP3 receptor-mediated depletion. It has been reported that STIM EF-hand mutants that are not able to bind catwall plus are constitutively active and continually activate SOCE independently of S/ER catwall plus in vitro and in vivo. The SAM domain is important for STIM oligomerization since mutants in this domain lack the ability to form inducible punctee. Catwall plus binding experiments in vitro using human STIM EF SAM residue 58-201 or STIM2 EF SAM residue 149-292 frang. However, STIM2 differs from STIM in that it is already partially active at basal S/ER catwall plus and becomes fully activated earlier during S/ER store depletion. Despite the same catwall plus affinity shown by STIM EF-SAM fragments, the full STIM2 protein showed a lower catwall plus sensitivity than STIM in transfected cells in vitro. This discrepancy indicates that other protein regions in addition contribute to the different catwall plus sensitivity or activation threshold shown by both isoforms. The hidden EF-hand domain does not bind catwall plus, but it is critical for intramolecular association, folding, and stability of the EF-hand and SAM domains. Very recently, it has been reported that structurally critical mutations in the canonical EF-hand, hidden EF-hand, or SAM domain disrupt catwall plus sensitivity due to the destabilization of the entire EF-SAM region. C-terminal region. Expression and tissue distribution. STIM2 mRNA is expressed by most human tissues. The STIM2 protein is expressed by many human cell lines together with STIM, indicating that STIM isoforms are co-expressed in the same cell, at least in the established cell lines. STIM2 protein is widely expressed in tissues, usually present at lower levels than STIM except in brain or liver, where STIM2 seems to be the dominant isoform. STIM2 transcription is also dynamically regulated, for instance being upregulated upon differentiation of naive T cells into THON or Th2 lymphocytes. Function. The STIM2 function has been controversial. Initial studies found that cRNA knockdown of STIM, but not STIM2, strongly reduced SOCE in mammalian cells. Liu et al. Reported a slight reduction in SOCE also by knockdown of STIM2 in HeLa cells. Sobolov et al. Suggested that STIM2 inhibits SOCE when expressed alone, but co-expressed with Orai causes substantial constitutive SOCE. In contrast, Brandman et al. Suggested that STIM2 could act as a regulator that stabilizes basal cytosolic and Cat-Wol plus levels. Parvez et al., using in vitro transient co-expression of human STIM2 and different SOCE channels in HK293 cells, reported that STIM2 mediates SOCE via two store-dependent and store-independent modes. Taking together, these results indicate a complex interaction finally regulated by the STIM STIM2, Orai cellular ratio and their endogenous levels. Studies performed in 2009–2010 using human, in vitro or murine in vivo models confirmed Brandman et al. Results and suggested that STIM2 participates in processes of the development and functioning of many cell types, including smooth muscle myoblasts, cells of the immune system, and neurons. Moreover, it is involved in tumorigenesis, the development of autoimmune diseases and mechanisms of neuronal damage after transient ischemic conditions. In resting conditions, cultured HK293 cells overexpressing or cortical neurons lacking STIM2 have increased or decreased resting intracellular Cat-Wol plus levels respectively, supporting the idea that STIM2 is essential for regulation of intracellular basal Cat-Wol plus levels. However, cells are very active in vivo and intracellular Cat-Wol plus levels are continuously fluctuating. The development of new methods to study the in vivo role of STIM2 in intracellular Cat-Wol plus levels would be necessary. In cultured human myoblast, STIM2 participate in cell differentiation into myotube. In the immune system, STIM2 participates in T cell activation, induced production of interleukin-2 and interferon gamma-F, probably by stabilization of NFAT residence in the nucleus, as well as in differentiation of naive T cells into Th17 lymphocytes, which presumably are important in early phases of autoimmune disease. In fact, STIM2 deficient mice showed mild symptomatology in the early phase of autoimmune diseases. In neuronal tissue, STIM2 plays a crucial role in ischemia-induced neuronal damage, and the absence of STIM2 in knockout mice reduced the neuronal damage produced by ischemia after transient interruption of blood flow in brain. This neuroprotective effect of STIM2 deficiency after an ischemic episode indicates that inhibitors of STIM2 function may thus have a potential therapeutic value as neuroprotective agents to treat ISC. Moreover, the same scientific study suggested an important role of STIM2 in hippocampus-dependent spatial memory, synaptic transmission, and plasticity. Finally, an oncogenic function has been demonstrated for STIM2, together with STIM in glioblastoma multiforme, where both proteins have increased expression and or increased copy number. Additionally, STIM2 is located in chromosome 415.1, a region implicated in invasive carcinomas of the lung, breast, neck, and head. Interactions. As mentioned before, STIM2 has been shown to interact with STIM SOCE channels such as Orai or TRPC, comedulin CAM, and also plasma membrane phosphoinositides. The expression of STIM2 has been shown to be influenced or regulated by presenilins in mouse embryonic fibroblasts and human B lymphocytes. References